we'll be going to our next speaker, which is Dr. Mario Masellis. He's an associate scientist at the Hurwitz Brain Sciences Research Program in Sunnybrook Research Institute. He's also an associate professor in the Department of Neurology at the University of Toronto and is the director of the Movement Disorders Clinic at Sunnybrook at the Department of Medicine. He is an Ontario Neurodegenerative Disease Research Initiative team lead, so that is ONDRI that we refer to, and he'll be discussing that in his presentation today. So please go ahead, Mario.
So thanks very much for giving me the opportunity to speak to you today. So thanks to URI and the C-OPN group. So I'm going to be talking about understanding heterogeneity in neurodegenerative diseases and using some examples from the Ontario Neurodegenerative Disease Research Initiative. So as you know, in terms of when we talk about late onset forms of dementia and Parkinsonism, they are complex phenotypes, and there are multiple potential targets that drugs can be developed for. So here on this slide, we have in the top left-hand panel, Alzheimer's disease, so your typical extracellular beta amyloid plaques and your neurofibrillary tangles, so it's a mixed amyloidopathy and tauopathy. The second most common neurodegenerative disease, as you all know, are the alpha-synucleinopathies, where we see these intraneuronal inclusions of alpha-synuclein in the form of Lewy bodies. And in the bottom half of this slide, we have the frontotemporal lobar degenerations, specifically the tauopathies, which can present both as Parkinsonism and or dementia or both. In the top left panel here, in the panel here, we see cortical basal degeneration with the cortical balloon neurons. In the next panel, tufted astrocytes typical of PSP pathology, and then the bottom panel, typical PIC bodies associated with frontotemporal dementia presentations. And then we have the TDP-43 proteinopathies, which not only cause FTD spectrum disorders, but also ALS. And there are multiple subtypes under this umbrella. Over the age of 65, there is a higher probability that you're going to have comorbid pathologies or mixed pathologies causing your decline. And specifically, for example, Alzheimer's and Lewy body pathology are commonly coexist in patients over the age of 65, and contribute to different phenotypes. So from Lewy body dementia to Alzheimer's disease to Parkinson's disease dementia. TDP-43 proteinopathies in the form of hippocampal sclerosis is also quite common and can be comorbid with Alzheimer's disease and other pathologies. It's also important, though, not to forget cerebral small vessel disease, another common pathology associated with aging. And this can co-synergize with the neurodegenerative pathologies to alter the phenotypes we see. So to date, we've had several clinical trials that have failed. Actually, the majority have failed in dementia and Parkinson's disease, although there's some promising ones that seem to be creeping up in Alzheimer's disease. And one reason why these clinical trials might have failed, in addition to just being, the drug not working, is that we are not accounting for the potential that these clinical syndromes are due to mixed pathologies, or in fact, the wrong pathologies. We don't always get it right. So patients, for example, diagnosed with Alzheimer's disease may in fact have, frontotemporal lobar degeneration presenting with memory loss. So we're off target. We're often also too late. So if we target patients that have full-blown neurodegenerative disorders, syndromes, then even if we could reverse the underlying pathology, it's too late. And so we have to push that bar earlier and earlier to get to the prodromal stages. We still do not fully understand disease mechanisms underlying neurodegeneration, and more work needs to be done focusing on that. And we also don't take into consideration a differential genetic background, which contribute to heterogeneity. So when we think about how we can kind of overcome these, I'll use the example of early onset forms of dementia, and this can equally apply to Parkinsonism versus late onset forms of dementia or Parkinsonism. Early onset forms often are due to autosomal, for example, dominant disorders, where we have genes of major effect. So these are monogenic disorders. We hope that the route to pathogenesis is a single route. This leads to more homogeneous pathology. We still see clinical heterogeneity, and this might be due to environmental, genetic, and epigenetic modifiers. And in this circumstance, the therapeutic approach of one size fits all might be the way to go. As I alluded to, in late onset forms of dementia, we don't have genes of major effect. We have several genes of minor effect identified through genome-wide association studies. These may all be, there may be different combinations of these minor genetic effects as opposed to kind of one effect in one patient, and different combinations in different patients. This leads to multiple pathogenic routes, more heterogeneous pathology, in particular mixed pathologies, clinical heterogeneity, which can be modified by the same modifiers, but also we must take into consideration aging factors and comorbid disease factors such as cardiovascular risk factors. And in this, we need to be thinking about therapeutics in a using personalized medicine strategies, as is done in the cancers with cocktail therapies addressing all of the underlying pathologies. So now I'm going to shift and talk to you about some studies that we've been doing in the Ontario Neurodegenerative Disease Research Initiative to try to kind of dissect out this heterogeneity and complexity to understand more about these complex mechanisms that yield neurodegenerative syndromes, and how this can ultimately improve our approach to clinical trials design. So ONDRI's mission statement is to catalyze advances in neurodegenerative disease research and practice that improve the diagnosis, prognosis, care, and outcomes for persons with neurodegenerative diseases and those that support them. ONDRI is a unique collaboration of individuals across the province of Ontario. Includes more than 80 clinicians and researchers, 35 research coordinators, 6 patient advisory groups, 5 project management team members. There are 10 core support sites which support all of our data platform captures, as well as 13 clinical sites for recruitment. And these are just some of them labeled, listed here. I'm going to be focusing on the first iteration of ONDRI, which, for which we have data collected and probably would be of most interest to this group at this point in time. And in terms of what we did was we collected several cohorts of patients: Alzheimer's disease, mild cognitive impairment, ALS, FTD, Parkinson's disease, in particular late onset Parkinson's patients, and those with cerebrovascular disease with or without cognitive impairment. The Parkinson's group included those who were cognitively unimpaired, those with MCI, those with dementia, so a full spectrum of disease presentation. They were studied across multiple different platforms. This included a gain and balance assessment, a genomics assessment, which initially focused on a targeted resequencing panel called ONDRI-seq, which looked at 80 candidate genes previously associated with neurodegenerative diseases, as well as the NeuroX chip platform. We also now have moved to genome sequencing in a subset of these patients who did not have identifiable variants on their initial ONDRI-seq platform. Eye tracking was collected, imaging data, including multimodal MRI data, volumetric T1, T2 weighted imaging, diffusion tensor imaging, SWI imaging, and resting state perfusion, resting state fMRI, as well as perfusion studies, ASL perfusion. They had clinical data collected, a full comprehensive neuropsychological platform, ocular coherence tomography for retinal imaging, and we've added in a pathology cohort where we're going to be getting autopsies on patients. And this also includes a molecular biomarker platform with Simoa-based markers such as NFL and p-tau181, as well as others, for example. Patients were followed at baseline assessments were completed and then had 2 years of follow-up. In terms of our initial subject recruitment, there were 520 participants. 161 were cerebrovascular disease, 53 with FTD spectrum disorders, 140 Parkinson's patients, 126 ADMCI patients, 40 ALS patients. Our retention over time was quite good in terms of a longitudinal cohort. We developed a very comprehensive data curation pipeline with standards and outlier detection, and then correction for any data that seemed a bit off. And then the data were released on BrainCode initially to ONDRI researchers, but now it's available, at least the baseline data is available to researchers around the world now through a data access platform. In terms of some of the research studies, I'd just like to highlight this paper, which was just published late last year, looking at the initial demographics of this of the ONDRI cohort, and just describing it in detail, which is important information to include in subsequent papers coming up. And now I'm going to shift and focus on two papers. These were both led by Tony Lang, looking at genetic and imaging modifiers of PD phenotypes using this ONDRI data set. So the first one was that I'm going to describe to you just briefly, just give you the highlights from, is Parkinson's disease, Notch3 genetic variants, and white matter hyperintensities. So in the ONDRI cohort, 139 PD patients contributed DNA samples. And in this cohort, 14 variants were identified within the Notch3 gene, which is the gene that causes CADASIL, mutations in which cause CADASIL. Now, these variants that we discovered were rare variants. They were non-synonymous. They were also potentially damaging variants, although we did not do any functional studies to show that they were damaging, but based on the literature that was out there, and also based on in silico assessments, tools that were used to determine potential pathogenicity, they all were potentially pathogenic variants. And so what we found in our studies that 13 individuals, 13 Parkinson's patients harbored these Notch3 variants. And in those that had these Notch3 variants, there was a doubling of the volume of white matter hyperintensity seen on T2 weighted imaging compared to the very negative Parkinson's cases. Across the whole cohort, we also found inverse correlations were observed between the white matter hyperintensity volume, general cognition, as measured by the MoCA, and essentially what we found was that higher white matter hyperintensity volume correlated with worse cognition. So just like to highlight, when we looked at, we weren't sufficiently powered to look at subgroup studies, but what we found was that in our analysis, there were larger effect sizes for this association between the positive patients versus the negative patients, whereby we saw a higher burden of white matter hyperintensity in the periventricular zone, as well as a higher likelihood that these were actually lacunes seen. So to summarize, this was the first study to report the presence of potentially damaging Notch3 variants in patients meeting criteria for idiopathic Parkinson's disease. And while these Notch3 variants are unlikely to cause a concomitant CADASIL in these patients, they may modify disease presentation by influencing the burden of white matter hyperintensities in Parkinson's disease. A second study that I'll just briefly summarize as well looked at small and large magnetic resonance imaging visible perivascular spaces in the basal ganglia of Parkinson's disease patients using this ONDRI data. And perivascular spaces are interesting for two reasons. First of all, they do represent small vessel disease burden, but they also may represent the glymphatic system, which is important for draining toxic proteins such as amyloid from the brain. And so when we looked at data, when we looked at small basal ganglia perivascular spaces, these are less than 3 millimeters in diameter, versus large basal ganglia perivascular spaces, which are greater than 3 millimeters in diameter, we found different associations with the data. So the small perivascular spaces were associated with measures on the part 1 daily non-motor symptoms of the MDS-UPDRS, as well as the subjective rated daily motor symptoms on the part 2. Whereas the large basal ganglia perivascular spaces were associated with the part 3 motor examination of the MDS-UPDRS, as well as motor complications. So we see differential associations based on the size of these particular, the volume of these particular perivascular spaces and clinical measures in Parkinson's cohort. So this study demonstrates how small vessel disease of the basal ganglia can contribute to clinical presentations of PD and potentially modify its course. I'd just like to shift and talk a little bit about two other studies just very briefly as well, looking at imaging biomarkers guided by genetic risk rather than clinical diagnosis. And this is using data from Sunnybrook Dementia Study, which has been led by Sandra Black for many years, and I've been involved in this over the last 10 to 15 years as well. And the reason for highlighting this study is that it's very similar to, it was kind of the originator, you know, of kind of where we got the idea to perform ONDRI, in that we looked at all of the major neurodegenerative diseases in exactly the same way with neuroimaging and neuropsych testing. As I alluded to, mixed pathologies can blur classic disease distinctions, and genetic factors may help explain these findings. And one genetic risk factor, the ApoE4 allele, which not only increases risk for pathologically proven Alzheimer's disease, but also increases risk for pathologically proven dementia with Lewy body spectrum disorders, as well as mixed Alzheimer's and DLB. So we wanted to look at the association between ApoE4 and hippocampal volumes in order to see if that may be one reason why the ApoE4 increases risk for both of these diseases and how it may do so. So I'll present to you some data from one of my former graduate students, Saeed et al., where we looked at ApoE4 and how it associates with hippocampal volume, learning, and memory across the spectrum of Alzheimer's and Lewy body dementia. So in this study, what we found was that depending on the number of copies of the ApoE4 allele you have, if you had 2 copies, you had smaller hippocampal volumes than those with 1 copy than those with no copies. And this was across all patients. And when we look at the breakdown based on Alzheimer's versus DLB patients, we see the same pattern. The only difference is that hippocampal volumes were smaller in the AD cases than in the DLB cases, which is what we would expect on average. We also saw a similar pattern in 38 neuropathologically confirmed cases. So this was one study that shows that hippocampal volumes may be an endophenotype by which ApoE4 can moderate the disease process underlying both DLB cases and Alzheimer's cases. A second study looked at ApoE4 white matter hyperintensities and cognition in Alzheimer's and Lewy body dementia. This was done by one of our former fellows, Syra Mirza, who's now a research associate with us. There's a complex relationship between white matter hyperintensities and cognition. And as you know, white matter hyperintensities are thought to be a marker of cerebral small vessel disease in most cases. They frequently coexist with the 2 most common neurodegenerative pathologies, Alzheimer's and Lewy body dementia. But cognitive performance clinically does not correlate necessarily strongly with the severity of white matter hyperintensity burden. So there may be some moderating factors that we don't know about. And these are just some cases whereby we have a patient who has essentially vascular cognitive impairment with, as you can see, a moderate to severe burden of white matter hyperintensity, who had preserved daily functions, and contrasting that with an MRI scan of someone who's more severely impacted, who also, and this person also had met criteria for dementia because their daily functions were impaired, and you can see there was a much more minor burden of white matter hyperintensities. So we wanted to understand more about why this may be the case, why there is this complex relationship between white matter hyperintensities and cognition, and it might relate to the heterogeneous etiology of white matter hyperintensities. They may be due to vascular compromise and ischemia due to cardiovascular risk factors, venous collagenoses leading to vasogenic edema, cerebral amyloid angiopathy, and Alzheimer's related pathology may also be contributing to this, or it could be a combination of all of the above. And in fact, we also have to consider that TDP-43, another neurodegenerative pathology, can also produce white matter hyperintensities, so not even vascular in origin. We wanted to study this by looking at also considering genetic vulnerability to neurodegeneration and ApoE4. So what we did was to perform a study to determine if ApoE4 modulates the association between white matter hyperintensities and cognitive impairment in patients with Alzheimer's and Lewy body dementia. And just the summary of this study, looking at data from both the Sunnybrook Dementia Study and the ADNI-I, what we found is that in E4 non-carriers, there was no association between cognitive factor scores on attention, executive measures, memory, and language, whereas in the E4 carriers, we found a strong association. So it might be that, a neurodegenerative genetic risk factor is modulating this relationship between white matter hyperintensity volume and cognitive performance. So in conclusion, I'd just like to state that in addition to the usual issues with novel drug development for neurodegenerative diseases, clinical, pathological, and genetic heterogeneity will continue to confound results, and it's mandatory to dissect out this heterogeneity using multimodal and deep phenotyping approaches to not only learn more about basic disease mechanisms, but also about predictors of rates of decline, drug responses, and adverse effects. And ultimately, we're going to need biomarkers for the different neurodegenerative pathologies, especially in the cases of mixed pathologies, to be able to unravel this. And I'd just like to mention my acknowledgements and thank you very much for your attention.